Frederick, thanks for the introduction. And I do want to thank the Boston folks who are present, that you came to my talk instead of, participating in all the activities that are going on today, the marathon and, doing your taxes and enjoying Easter and stuff. So today I want to talk about our image processing pipeline. And just to maybe set the stage, I'd like to start out with this table that was published in a recent review that highlights just the overall explosion in the number of spatial omics technologies that we've seen. Without question, single cell sequencing has revolutionized our understanding of biology. And I think in the last years, there has really been this push to take it a step further and to introduce spatial information at single cell resolution. And so typically, the technologies are compared by coverage and spatial resolution. And so you probably see some of your favorite technologies here. So the tables may be a little hard to interpret, especially the spatial resolution. So there's some margin notes over here is that typically when you see something like 20x optical magnification, that often corresponds to single cell, and things that are higher correspond to subcellular resolution. Super resolution, if you're not familiar, this is just a diffraction limit of the light, and you really need to go to something like electron microscopy if you want even more resolution than that. And then, comparing that to single cell data, so epithelial cells usually are around 25 micron, immune cells are a bit smaller. And then one thing that this table doesn't highlight is there's two other dimensions that I like to think about when talking about these technologies. And one is cost and effort, and this really determines the amount of throughput that you can do when processing data using these technologies. And a good indication that something is a scalable technology is whether or not there's a commercial entity backing that technology. So, as to give an example, Codex, for example, is backed by Akoya Biosciences. That's really trying to take it to industry scale processing. And then the other thing is tissue preservation. And there is an interesting mix of some technologies where you might get a low-plex imaging of the entire whole slide, and then identify important regions of interest in that slide, and then go in with a deeper sequencing technology, something that gives you higher coverage, but maybe lower spatial resolution. And so we're kind of seeing an emergence of these hybrid technologies that will require additional possibly innovation in the tools to process that type of data. So in our lab, we work with CyCIF, or cyclic immunofluorescence. I think most people are probably familiar with this technology, but the idea is that we do cyclic acquisition of low-plex images. So in every cycle, we stain for 3 antibodies. We acquire an image just using standard microscopes. Every cycle will also have a nuclear stain, so identifying where the cell nuclei are. And that's really what allows you to then register your images across the different cycles. So one can imagine in this example, we collected 5 cycles with 3 antibodies each and a nuclear stain. The nuclear stain we use to register them, which then gives us 5 times 3, 15 channel image in addition to the nuclear stains. And just to kind of give you an idea of the types of images that we're collecting and the scale of the data that we're collecting. So I've reached out to some of my experimental colleagues and just ask them, tell me what are some of the large images that you've collected past year, and these are some of the statistics that they send back. So some of the largest XY coordinate, some of the largest images in the XY dimensions we've seen are close to 70,000 pixels on one side, and we've also been able to push really the number of channels that we acquire. And so, one of these images is easily 1/4 terabyte. So you can imagine that a full data set will be multiple terabytes. So this really presents, I would say, a challenge for scalable image analysis and for processing it. And that's what motivated us to come up with this image processing pipeline that can hopefully help us push a lot of this data through in an automated fashion. One other thing I want to mention here, so this is a collaboration with Rareside, but we've been developing what we've been calling an Orion platform. What this lets you do is collect 21 channels in a single cycle image. And probably around 15 or 16 of those channels will be fixed. And then the last 5 or 6 channels will allow you to select a panel of markers that are important for your particular disease. And the big advantage of this panel is, and I might get into it a little bit later, is that if you build some sort of a predictor, say a cell type classifier that works with your 20 markers that you've imaged, somebody else comes in with a different panel of 20 markers. So how do you apply the model that was trained on this marker panel to a new image that may or may not have the same markers? And so this is our kind of one of our efforts to try and standardize the set of markers that are available, which will hopefully help standardize the image processing as well. So Ludwig already briefly alluded to, so we developed a pipeline we call multiple choice microscopy, or MC Micro. We recently did a massive revamp of the website, which will hopefully is now more user-friendly. Originally it was tailored more to the reviewers, just to make sure our paper gets through and that the reviewers are happy. But this is now should be a lot more user-friendly. So the pipeline, individual modules in the pipeline have been containerized using Docker. And the reason for that is, so our first iteration was focused on using Conda environments, and it's been a constant source of problems and constant source of friction, where it was a lot of, well, it works on my computer, right? And maybe on your computer, it doesn't have a certain set of libraries. So this has really been our push to standardize a lot of our modules and to make sure that they're distributed as Docker containers, because it simplifies execution across the different environments. And a lot of our end users are experimentalists. And we all have our favorite languages. So I'm a big fan of R, for example. But a lot of different modules could be written in different languages, and we can't really have experimental users maintain a Python environment, an R environment, a Java environment. So our solution was we teach them how to run Docker containers. We provide command line interface to all of our tools, and suddenly the experimentalist is able to run any tool under the sun that has Docker and a command line interface. So that's been our main motivation. And then these containers are strung together using a workflow language called Nextflow. And what this language really lets us do is define how the data flows through the pipeline. And that definition is independent of how it's going to be executed. And the execution is controlled by several different configuration files. So there's one for running it on a local workstation. We have a separate set of configurations for the O2 cluster. And then we've successfully also executed on the Amazon and Google clouds. So we've tested this with data sets collected by these platforms, so not just SciSIF. Yeah, so I'll step through this pipeline here in a few minutes, but I also want to point out that we've formalized the definition of data levels to really maybe mimic what's happening with the sequencing data. So, you might be familiar if you're working a lot with sequencing data that, you have your raw reads, and that's, level 1 data, and then that becomes aligned to a reference genome, that's level 2, and so on, all the way down to level 4, which are your single cell sequence, single cell feature tables, right? Where every row is a cell, every column is, say, the expression of a marker. So we try to do kind of follow the similar workflow, and the formalization of this data levels is available as a separate effort, which we call minimum information for tissue imaging, or MITI. We've also partnered up with our collaborators at Oregon Health and Sciences University to also bring MC Micro to Galaxy. The main advantage of this is that it allows users to specify how the data is connected or how it flows through the pipeline using a graphical user interface instead of a command line. And some experimental users really find that to be a lot more intuitive than typing commands in the terminal. But because the containers are Dockerized, it's been a pretty straightforward, just plug it into a different workflow. We've released a couple of data sets to go along with MC Micro, and this really is meant to help additional method development and testing of methods and benchmarking them. And so we expect that these data sets will grow over time. But presently, there's one tissue microarray, which contains 123 cores that span 34 different tissue types. And then there's several whole slide images. It's the same specimen that was serially sectioned. So it's a tonsil specimen that was serially sectioned, and the different sections were sent to different institutions, including here at HMS, Oregon Health and Sciences University, and the Broad. And each institution acquired images using their technology. So we have SciSIF, multiplexed IHC, and codecs. And this really lets you compare how, it lets you run the pipeline across all the technologies, but it also lets you compare the outcome of the different technologies. And you can dig into that and see the different advantages and disadvantages. Okay, so what I'd like to do now is maybe step through the different steps in the pipeline and really maybe highlight specific, certain features of individual steps and maybe properties of images that it's good to pay attention to as your data makes through the pipeline. And the processing really starts with what we call illumination correction. So if you collect data using traditional microscopes, the outer edges of a tile will often be dimmer than the center of the tile. And if you were just to take those tiles and you stitch them together, what will often happen is you'll see this checkerboard pattern. This is a telltale sign. Now, this is obvious problems for downstream analysis, because if you have a pixel that's artificially dim, you might interpret that, oh, a marker is not expressed, whereas in reality, it's just hasn't been corrected for illumination. So there's a really nice method that's been published out there. It's an ImageJ plugin. ImageJ is a popular viewer for images, viewer and analysis tool. So what we've done is we've taken that method and we containerized, and it's available to run as a standalone tool, not even necessarily as part of the pipeline. And what it does is it corrects this checkerboard pattern so that the images come out to be with even illumination. So another important aspect is typically the field of view of a microscope is not wide enough to collect whole slide image at once. So what often happens is you'll image a section of an image, a section of a specimen. You move your microscope stage over, image the next tile, and so on. And so what you end up with is a series of tiles that have a little bit of overlap between them. So the first step before you can do all downstream analysis would be to stitch those tiles together in XY coordinates. And then remember, this is also a cyclic process where you stain for 3 markers at a time in every cycle. So you might have, 3 to 20 cycles. So you need to do both stitching and registration across the cycles. So we developed a tool called Ashlar that does both steps simultaneously. And we found that it works better than doing stitching first and then registration, or registering and then stitching together. So trying to do both steps at the same time. And that's, again, available to run as a standalone tool. When working with tissue microarrays, we've also developed a tool that will identify the individual cores and images and then split those out so that each core is a separate image. And what this lets you do then is parallelize all downstream processing. So if I have 123 cores, then I just spawn 123 processes and they all run in parallel. And yeah, so I mean, that's, an obvious advantage there. This is a really clean image of a tissue microarray. In reality, that's not always what they look like. So here's an example where, it's not a rigid grid. Some cores are off to the side. Some cores are not really, full circles. So what we actually ended up doing is using a popular deep learning architecture called UNet to go from images like this to really identify where the presence of cores are. So, it might seem like a trivial task, but in reality, there's a bit of deep learning happening in the background that does this, that then annotates individual cores. Now, I would probably say by far the #1 issue right now that comes up is that people's machines run out of RAM when they try to process these giant images. And we've started to take steps towards splitting up an image and processing individual pieces separately, again, in parallel. And the first step to that was to adapt choreograph, where instead of extracting individual cores from a tissue microarray, we extract chunks if there are multiple chunks present on the slide. And then those, again, are written into separate files, and the files will be smaller, so it'll not require as much memory and not have as much problem. But this is still, I would say, an open issue, and we have an open GitHub issue right now where we're also thinking about, well, what if you don't have tissue chunks? What if it's a single piece of tissue, and you really need to split it up in a way. And turns out that it's actually not a trivial task, because you want to split it into strips. The strips have to overlap, but you have to have a special handling of cells on the boundaries between the strips. So there's, that's still in progress. So then, speaking of segmentation, right? So then once you've built up your images, so either you stitch them, align them, maybe partition them using choreograph. The next, and I'll probably say this is the key, most important step, is identifying where individual cells are in images. So what MC Micro does is it currently incorporates a homegrown method called unmixed. We have elastic segmenter from the Broad. We have Mesmer, which was this really popular method from last year, I guess, or 2020, from Van Valen Lab. And we're also hoping to soon incorporate CellPose from the Janelia. And the pipeline will run all of these methods in parallel, or whichever selection of methods you tell it to. And the advantage of that is that this then lets you downstream look and decide how the methods compare to each other. Is one better suited than the other? So here's I'm showing an example of our homegrown method, unmixed versus elastic. We see that unmixed is able to find smaller cells a little bit better, and elastic might need more parameter tuning to be able to find those cells. A big kind of advantage of the pipeline is I really try to work hard to make it so that if somebody wants to add a new module, they don't have to touch the Nextflow code base. And the way it's implemented right now is that there is a configuration file available there that anybody can edit. It's a plain text file. And the way you specify modules is you say, what's the container that needs to be executed? What is that container Docker tag or version? And then what is the command that you need to run inside that container? How the module accepts the input, whether or not there's a custom model. And then if you're familiar with segmentation, whether or not a module also requires downstream watershed. And so the pipeline will parse this configuration file and assemble the commands using this information behind the scenes. So really, there is no Nextflow editing necessary. So I just want to maybe say a few words about how our homegrown segmenter was built. So we really made an effort to pull together data sets, training data from a number of different tissue types. And the reason for that is, you can readily appreciate that some tissues have densely packed cells, and it's a much harder segmentation task than something like, fibroblasts here in this example. So this was kind of one of our important aspects, is just pulling more tissues together. The second one is we used a nuclear envelope stain. This is a lamin stain. And what this lets you do is, so contrasting to DNA, it really highlights kind of the edges of the nuclei rather than the nuclei themselves. And this makes it a slightly easier problem for the downstream segmentation. And then, we did our traditional data set augmentation where, in focus, out of focus, shift translation, and all the usual stuff. And what we've noticed is that by combining multiple tissues, data augmentation to do blurring and other effects, and nuclear envelope stain, we're really able to push the performance of segmentation over just traditional DNA-only segmenters. The way you read these plots is, so this is analogous to ROC curve, where on the x-axis this is the level of overlap you look for. So for example, you're looking for 70% overlap between your predictions and ground truth. And then the y-axis is what percentage of cells satisfy that overlap or better. Now, somewhere here would be a vertical line that corresponds to human level performance. Let's say if you put me and Ludwig in a room and you ask us to annotate a data set, we're not going to have a perfect overlap in our annotations, and there's going to be some 90-95% overlap. And so you generally, ideal predictor would be 100% up until that vertical line, and then it doesn't matter what happens after that, because even two humans can't agree. Okay, so just working our way down the pipeline. There's, we've been in the past year or so, we've been making a lot of effort to standardize QC and to systematically define the different artifacts that happen. And this is an effort that's been led by a postdoc in the lab, Greg Baker. So he built a human in the loop QC tool. So behind the scenes, it uses this Python-based Napari image viewer, and it lets a user to inspect their data, both the raw pixel values as well as the summary distribution of marker intensities, additional UMAP clustering and so on, and just interactively modify what portion of the data should be excluded. So there's a lot of different moving screenshots here, but here's one example that you might choose that to exclude overexpression here for whatever reason, like you decide that that's an artifact, maybe it's antibody aggregate that just happens to shine brightly, and so you want to exclude that. And the effect that it has is at the end of the day, it writes out your table excluding cells or rows that corresponded to artifacts. Now, this module is not automatically executed by the MC micro pipeline, and the reason for that is because it requires human interaction. But the output of MC micro and the SciLinter input are all meant to synergize. So it's, you could just open SciLinter, point it to MC micro output, and you'll be ready to go. So no additional wrangling necessary. Now, Greg has really, and we'll revisit this towards the end of the talk when I talk about future directions, but Greg has really gone through and systematically tried to define the different types of artifacts that might be present. And one can already think about some of this could be easily detected using automated tools. And this is where we're going now, is we're hoping to design a set of predictors that will automatically find these artifacts in the images and allow for their exclusion downstream. Now, others, things like, under segmentation or over segmentation is not trivial to detect automatically, because your segmenter is already an automatic tool that's meant to extract cells from images. So it's sort of a, catch-22 is how do you know how well it's doing without human inspecting it? The other thing I want to say about this is there is hope that some signal can be recovered from an artifact. I'll give you an example. So a tile stitching error. So this is where you had two neighboring tiles that somehow didn't get aligned properly. So one can imagine that this could be fixed in post-processing, and the data that exists there will be recovered. Conversely, if you have something like an antibody aggregate, which completely covers your image, there is no hope to extract the underlying cells. So that's just, we would need to exclude that area from the analysis entirely. Okay, and now, making it all the way down to the bottom of the pipeline is the quantification module. So what it does is it takes your segmentation mask, it overlays it on top of the original image, and then using the areas in the segmentation mask, it then quantifies the features of individual cells. And again, what comes out are these single cell tables that we're all used to working with, even from other modalities. Every row is a cell, every column is a feature. The features will typically be either marker expression or morphological features such as, perimeter or area, or, how elongated the cell is. And of course, there's, the XY coordinates. And now these tables are ready to go into your favorite single cell analysis tool. I think downstream, it's fairly well developed and fairly well standardized. We also have some homegrown tools, of course. One is SciMap, that's led by Ajit Johnson in the lab. So this is really meant to bring some spatial analyses. And we heard this wonderful SquidPy talk from a couple of weeks ago. So this is an alternative to SquidPy, I would say. And the underlying methodologies is really based on this topic modeling from natural language processing that's been around for decades now. The idea being is that you have an article, and the observations are your words, and words could be used to assign parts of the article to different topics. Like maybe you're talking about sports here, and you're talking about weather here, and so on. So this is a similar idea where your marker expression are your observations, and your topics are some spatial arrangements that are recurring somewhere. So Ajit defines this term called recurring cell neighborhood. So this is a particular arrangement of cells that repeats in the image. And so he uses this idea of topic modeling to identify where those arrangements exist. And of course, there's tons of visualization. I think I saw Niels is in the audience. So we're hoping at some point to take MC Micro and bring the test to it so that maybe we can automatically generate some dashboards to let users inspect the outputs. I know that our Galaxy friends at OHSU have already done that. So they've inserted the test into their Galaxy dashboard. And so we'd like to replicate that on the Nextflow side as well. But in addition to existing visualization tools, we also, again, have a couple of homegrown ones. So if you were to go to scisf.org right now, you'll notice that there is this interactive storytelling that really guides you through the different areas of the image. And this is powered by a tool that we developed in a lab called Minerva that consists of two parts. There's the Minerva author, which is meant for people who would like to design and write these stories and identify that, say, here's an interesting region of my tissue that you should look at, and here's what's happening in that tissue. And the second part of that is, of course, Minerva story, which then takes that information and presents it as a visual, interface that lets a user step through it. So these tools are open source, available for visualizing any images. If you go to mcmicro.org, there's one tutorial that steps you through all the steps I just talked about, that shows you how the images get stitched and registered, how the cells are found in the images, and so on. And it uses Minerva story to guide you through the different parts of the image to do that. We also have a collaboration with the Visual Computing Group at Harvard Computer Science Department. And so they have been developing this tool called Scope to Screen that enables visual analytics or interactive viewing of the image while also performing some sort of analysis on top of it. And they developed this tool that they call lensing, and it has an advantage of allowing you to display some contextual information for individual cells while also having the broad context of the image as a whole. So here I'm just showing several examples. One is you could just look at the distribution of marker intensity. So you put this lens somewhere in your image and it tells you, well, it's expressing the following markers with the following intensities on average across this area. If you have cell type annotations that you derived using your downstream tools, you can also overlay that information on top of that. If you have multiple imaging modalities, so if you're looking at your scisf data and you also have matching H&E image, you can overlay that, and this lets you look at multiple image modalities at the same time as well. And of course, you can throw in some machine learning behind the scenes as well. And so they also built this tool called HistoSearch, where it tries to identify regions that have similar expression patterns and morphological patterns. And then they'll cluster them so that when you highlight a particular area in an image, it'll also highlight other areas in the image that have in some way similar properties. So I encourage you to look at their Scope to Screen GitHub repo, and it'll have a few more examples. Okay, and then I'd like to spend the next 10 minutes talking about our maybe long-term vision for where some of these things are going and what we're thinking about. And there might be an opportunity for some collaborations with members of the audience. So I just want to mention that I am a co-chair of the image analysis working group for one of the consortia that we belong to. And this has allowed me to keep tab on, I would say, the pulse of the image analysis as a whole in the entire field. Because we're doing a lot of surveying of existing technologies and methods. We have invited speakers every month. And we've also been doing these image analysis hackathons. So we did that looks okay. Sorry, I'm just gonna stop here and switch to this slide. I unskipped a slide on my other computer and it didn't trigger here, but I just wanna point out, so we we started doing these hackathons actually right before the pandemic hit. So you might notice that this is a week before we all went into lockdown. But we got together in January, and this was still at the time when everybody was thinking about things like segmentation and registration, so very basic image analysis tasks. We systematically defined the data sets to be used for those, and then we hosted a hackathon, and this has now been written up and published. But just to give you a sense of how fast the field is moving, so this was 2 years ago. By now, I would say there are existing solutions, both in our lab and elsewhere, that do all these basic image processing tasks, so registration, segmentation, quantification, clustering. So what are we thinking about now? So what we did is we partnered up with members of H10, HubMap, and there's a couple of smaller consortia as well, and we tried to define some image analysis challenges that are relevant today right now. And so we hosted another hackathon. This one was all virtual in February. And there were 11 challenges, and I just want to break them down into 4 themes. So by far the biggest theme was handling artifacts. So I already spent a bit of time talking about this, but there was a challenge on building automatic detectors of artifacts. There was a challenge of, if you now have an accurate detector of artifacts, how do you use it to make your segmenter more robust? So how, make sure that it excludes the artifacts, or how does the segmentation become more accurate? I'll spend a little bit of time talking about this. This is actually maybe not the most well-known problem, but we've noticed that it's surprisingly prevalent. And chances are, if you're working with the data, you have this problem in your data set. And then there was a challenge for removing the artifacts, and this was purely for cosmetic reasons, because nobody's going to believe you if you just impute the data that's behind an artifact. So this was entirely just to draw attention away from the artifacts when you present data. A couple of other things that I unfortunately won't have time to talk about, but there's a lot of interest in image translation right now. What I mean by that is, if you have an H&E slide, which is present everywhere, in every hospital, what would that tissue look like if you were to image it with SciSIF? Now, there's several methods right now that have been trained using these serially sectioned data sets. So you can imagine taking a specimen, say colorectal cancer in our case, serially sectioning it, imaging every other section using H&E, and then every other of that using SciSIF. So now you have neighboring slices with different modalities. And you can train your deep learning network to predict what SciSIF looks like from H&E. Now, it's not perfect, right? Because there's some 5 micron difference between them, but it's usually close enough to get a good result. There's been a lot of methods that do pixel level analysis, and I'll get to it in a moment. And so this is complementary, I would say, to our traditional single cell feature tables that we're used to. Here, the variational autoencoders or any other type of things are working directly with the n by n pixel windows, so pixel level features. Thumbnail generation, so if I have an image of breast cancer tissue, how do I collapse my 40 channel image into a nice thumbnail that highlights the most important properties of my specimen? So typically, if you have a picture of a cat or a dog, you only have 3 channels, so deriving a thumbnail is trivial. If you're working with 40 channels, it's not trivial. So that was the channel there. We had a couple of visualization challenges, including 3D visualization, which again, because of the serial sectioning and multiple images in the Z-planes, this is now an important aspect, is how do you navigate 3-dimensional data? How do you visualize it effectively? So breaking into that dimension. And of course, I think I alluded to before is, this is terabyte scale data. It's an order of magnitude higher than a lot of the sequencing data. So there's some non-trivial things that must be handled to be able to run this at scale. And we're, I just want to mention that we, constantly pushing the memory limits of O2, and, certain images will request 230 gigabytes of RAM, and they'll still, run over. So just want to highlight again that we're hoping to move towards automatic detection of artifacts. Again, this is led by Greg Baker. So he has painstakingly went through and annotated artifacts in one of our data sets, and then gave that over to the challenge participants, who were then invited to make prediction whether individual cells are parts of an area that's impacted by an artifact or not. And then, from that, they were able to compute some prediction statistics. Okay, so spatial crosstalk. Yeah, so I really want to highlight this because I think chances are you have this in your data and you may not even realize it. And what happens is, if you have a surface marker, so it's a protein that's expressed on the surface of a cell. If you were to image it, chances are that signal from that channel may appear on a boundary with another cell and may incorrectly get assigned to that cell during quantification. So just to remind everybody, what quantification does is it'll take a cell segmentation mask and it'll usually average the expression of a marker. So if a marker is expressed on the surface of a cell, it's kind of an average over the entire surface area. And if it's neighboring nearby cell, that signal might bleed into this cell, the index cell. Now this creates two problems. One is you might get a nonsensical combination of markers. So if we were to do quantification, then suddenly this cell expresses CD20, CD3, and CD68. Well, that's not a combination of markers that you would generally expect to see here. And the other problem is, if you're now to do clustering on the single cell data, you might get cells incorrectly assigned to clusters they shouldn't belong to. So there's this paper that I first identified this problem. They also proposed a method to correct for it, which, during quantification, what you do is you adjust the boundary towards cell A or cell B, depending on which cell you believe actually expresses that marker. And then you fix the quantification so that only the signal that belongs to that cell is quantified. Now, there are original implementations in MATLAB. We took it, we moved it to Python, we containerized this method, but I would probably say this is still an area of active research, because our experience has been somewhat mixed with whether or not this method works well. We've actually noticed that sometimes the boundary ends up moving in the wrong direction. So instead of, if there's signal bleeding away into nearby cells, instead of that signal being collapsed back to the central cell, somehow it'll adjust in the opposite direction. Even more signal will come out of that. So this is not by any means a solved problem, but I just want to highlight how often it happens. So here's an example where it's a CD8 positive T cell that passes through the tumor epithelium. What I'm showing is the outlines of the segmentation mask. Then I'm showing the three markers. So CD8 is the marker of the immune T cell. Keratin and E-cadherin is the markers of the tumor epithelium. The little white dots are cells that have been assigned to the same cluster. So, you would interpret it as it's a cluster of cells, of T cells, right? But because of this signal that leaks into nearby cells, those neighboring cells also get assigned to that cluster incorrectly. They shouldn't belong there. So this is, a telltale example. If you were to look at your clusters back in the original image space, you'll notice it's a cell and the neighboring cells that got assigned to that cluster. And this is not correct. So we need to be able to fix that. Just to, again, show you how prevalent this is, here's a really clean clustering solution, right? So this is a common thing to do. You have your single cell feature table, you cluster it, present it using UMAP. Here's a cluster 11, well-isolated cluster, no intermixing with any other cells. It's a cluster of B cells. If we now go and look at where the images, where in the images those cells happen, same problem, right? This is, again, a telltale sign. CD20 is a marker of B cells. Here's a B cell that expresses it. Some of its signal leaks over into nearby cells, and they also get assigned to this cluster 11, even though they shouldn't be in that cluster. So again, I think this is a lot more widespread and prevalent than people realize. And I think we need a good solution to be able to fix it. So we've actually been thinking about moving away from spatial feature tables towards pixel level analyses. And we kind of envisioned that the next iteration of tools or the next generation of tools will not just work with spatial feature tables or single cell feature tables, but also bringing these image patches that contain actual raw pixel information. So, this is a canonical workflow. Is once you have your spatial feature table, you'll do some filtering or QC, and then you feed it into your UMAP, latent derision analysis, whatever. But what happens is you lose a lot of important information that was present in the earlier. So cell-cell interactions, cell morphology, spatial distribution of expression. So I don't know if you can appreciate it in this image, but sometimes markers will get polarized. So when you're collapsing information about marker intensity into a single number, you lose this polarization information where maybe in this case, CD3 was polarized to one side of the cell, and maybe that has something to do with the cell type and cell state, and you lose that information. So in our view, you actually want to maintain the original raw pixel intensities and use that in parallel with your spatial feature tables using your favorite deep learning methodology. So to do that, we built a tool called Cell Cutter. So it'll take the output of the entire pipeline, and what it'll do is maybe I'll just jump to the next thing. It'll cut out individual cells, optionally masking out the background. So if you only care about the cell itself and not the neighbors, you can choose to do that. Or you can tell it, give me that cell in the center and all of its surrounding context. And so what you get back is an n-by-n pixel patches that are all stored. So like here's an example of 46 by 46 patches. We extracted 9,000 cells across 12 channels. They're stored in a ZAR array, which then lets you feed it directly into your deep learning methodology. Okay, so I will stop here and I'll open time for discussion. But I covered a lot of different tools, and I just wanted to show a summary table. And I'm happy to share the slides as well, Ludwig. And this just allows you to go directly to the GitHub repositories and explore the individual tools. We are hoping, so we just finally secured some funding, but this is a huge amount of programming and a huge effort in general. We're hoping to hire a scientific programmer to help with that. This is not an official ad yet. We haven't put it out. Hoping to do so within the next few weeks, but if you know somebody who's looking for a position to do open source development, please let me know. I would probably say the most mandatory qualifications, we'd like somebody who already has some hands-on experience, either through Bioconductor or other projects working on large-scale open source software projects, and we can teach everything else. And I just want to acknowledge all my collaborators. So this is obviously a huge amount of effort done by dozens of people. I'd especially want to thank our experimental colleagues and early adopters who really took MC Micro when it was first developed in 2020 and help us catch a lot of different bugs and to nail down the issues. And this is still an ongoing development, obviously, but all of these people have now been using MC Micro on a regular basis to process their images. So I'll stop here. One last thing I want to mention. So we do monitor the imaging forum. So if you have any questions about the pipeline, you can obviously go to GitHub and just open an issue. But if you want to, you can also post your question on the imaging forum, just tag it with MC Micro and somebody will reach out.
Yeah, excellent. Thank you so much, Artem, for a really great talk and for leading us in such a structured and easy to follow along way to what I think is a quite complex pipeline. I also really appreciated, if I maybe start off the question and discussion session, but people really should feel free to raise a hand or put question into the chat, but maybe I really also appreciated this figure 1 pipeline, which already was quite instructive when reading the paper, where you kind of like laid out the individual pieces and modules, but also what I found very helpful, these individual data levels. And I like that you kind of like made this analogy to RNA-seq, because that was the first thing that came to my mind, TCGA data levels, right? Where you have maybe your FASTQ raw sequencing output, and then you have your BAM alignments, and then you go over to your raw or normalized reads. Now, when I do RNA-seq, often enough, these data levels are not really like, intertwined. Typically, if I have my BAM file, I do some analysis on the BAM files, I'm not going back to these FASTQ files anymore. Or if I have my raw read count matrix and I go over to DSC2 and to do my differential expression analysis, I do not care much about these BAM files anymore. Now here, this seems to be quite different, right? I mean, I wonder whether you can comment a little bit more on these back and forth and these interactions between these data levels. You kind of went there already, like in the last slide, which I found very, very interesting, because I'm just wondering, if I arrive at this spatial feature table where you're kind of like your last slide said, well, maybe that's not the solution in the end. How much do I still need to carry around these big images and these individual tiles, or do I need them not really anymore once I have arrived at my spatial feature table?
So I would say you definitely don't need level 1, because all of that information will be present in level 2 and it will be stitched and registered. But you're right. I mean, you raise a very important point, and I think this is where imaging data varies from sequencing data. And that is, because those are images, you almost always need to go back and look at what's actually happening in the image itself as you're doing your single cell table analysis. And for better or for worse, we as humans are pretty good at finding patterns in images. Like, I could look at some of the examples that are shown here, right, and immediately recognize that there are some cells that got assigned to a cluster that shouldn't be there. They're there because they're neighboring a cell that we care about. This is not something that can be easily picked up from a feature table, right? But it instantly jumps out at you when you look at the images. So this is, again, for better or for worse, you do need pixel level data. Now, there was some concern when we were putting together this mighty definition of data levels, is that NCI, as you say, they typically don't care to preserve level 1 or level 2 data that might go into, cold storage or glacial storage, right? That, you may never access again. So we've even played a little bit of this game that maybe level 3 is not just a segmentation mask, but maybe it's a whole slide image that has undergone some form of QC, but you still have your raw pixel information, because it is important for some of these analyses. So yeah.
Excellent. Yeah, that makes a lot of sense. Are there other questions from the audience for Artem? I see that Katie asked in the chat whether these slides could be made available, which I think
I'm very happy to share the slides. So maybe I'll just email the link to Ludwig.
Excellent. And we put it on the side. So that will be great. Yeah, maybe I ask another question out of personal interest in this stitching that you're doing with Ashlar, if I pronounce this Ashlar, if I pronounce this correctly. So that seems to be super helpful also for data that I'm working with. I wondered how technology dependent is that actually? Can I put in arbitrary multi-channel TIFFs, or would that need to be some sort of sieve?
So the important aspect here is that the data needs to not already be stitched. So we've had some people reach out to us who only want to do registration, right? So you've already stitched your tiles in the XY coordinates, and now you want to register your cycle 1 to cycle 2 and so on. We have a separate tool for that. Ashlar will not work well. And the reason for that is because it does affine, or I guess rigid, right? Transformation is what it would be called, where it doesn't warp the data. So if you have stitching that doesn't quite line up across cycles, then it'll just generate errors for you. So if your data is not stitched, use Ashlar. If it's already stitched and you just want to register it across cycles, I can point you to another tool that we have for that.
Excellent. Yeah, no, I have these tiles. So these are C-stack TIFFs that I think one want to stitch together, and I will certainly try this one out. What do you actually mean with registration in this context? Is it just that you register the first level, that you say okay, I have 800?
Each cycle here will be 3 channels, right? So you have your tiles. Each tile will have, as I'm showing here, 4 to 6 channels, right? So to get your final 40 channel, 60 channel image, you need to line up those tiles across the different. It's almost like, imagine if you were to collect red, green, and blue separately.
Mhm.
Now you want to line up the colors in addition to stitching tiles in the XY coordinates.
Mhm. All right. Other questions for Artem?
And I should mention that there is a number of parallel efforts that try to do this with non-rigid methods as well, which is helpful if your specimen somehow shifted or rotated or maybe folded as the cycles progressed. So like there's some non-linear transformation that happened to it.
Mm.
And those methods tend to, as everything else these days tends to be deep learning based. Whereas Ashlar is kind of more canonical. It'll take an image, transform it to frequency space, and then do line up in the frequency domain, and then transform back to the original space. So it's more classical image analysis. Tyrone, right, has a question?
Yeah, Tyrone.
Excuse me, sorry. So I'm really interested in some of the UNet implementation that you used right there. I'm curious, since you have everything containerized, are you using the UNet for various different segmentation stuff? Are you using the same module for each one, or are you tweaking each module to use a
different models for different tasks? And this is actually maybe a parallel question of interest to me, which is how to distribute models. Like say Ludwig trained his own model that does something better. How do we take his model and insert it into our container, right?
I was about to ask about that because because a lot of the times that in my experience, a lot of the deep learning models require a lot of tweaking, especially if you're using in a very particular context, like choreograph here. So I'm wondering if you're using the same weights for the entire workflow. But
so it's the same weights for a given task. Yeah, so there are, unfortunately, models are not tweaked to a particular data set. Like, so if I'm doing de-arraying of a tissue microarray, right, if I'm identifying the individual cores, it'll be the same model applied to every tissue microarray. But it'll be a different model than the one that identifies large tissue chunks.
So in your experience, would it be more useful to train for your particular data set rather than use the generic?
Yeah, so we have some experience of that with Elastic. So that's the broad cell profiler segmenter. So it's a random forest based method, but they have a graphical user interface where you can pull up your image, you can annotate where the different classes are, and it'll train a model, and you can provide that model directly to MC Micro. And we've seen, I would say mixed results with that. So sometimes it obviously does better, because if your data is particularly peculiar and there's some certain properties that are not represented in our standard models, then yeah, having a custom model totally helps. The downside of that is, that's a random forest model, and it's, still has some limitations, and it doesn't usually do as well as deep learning based models over bigger data sets. So it's, we'd like to be able to allow similar things happen on the deep learning side, but we don't have the infrastructure. And to me, it's an interesting question of how you would even go about implementing that, because I see this in the field of machine learning in general, is that typically it's a one-shot model, right? So you train it once, you deploy it, and then it's ready to go. And it's not always trivial how you would go about updating it, or how somebody else would bring in their model into that.
In many cases, when you're trying to deploy, yes, it's just that you would probably have a lot easier time getting rid of crosstalk if the models were particularly tweaked, because if you have a certain data, if you can get a certain data set, usually would optimize in order to reduce that level. You can tweak it, the hyperparameter, such that you won't get such false positive, but you probably lose a lot of data in that case. So it's I'm just wondering how fluid it was, but it looks really good. At least you're using Nextflow to separate things out, so it's very impressive.
Thank you. So we're at the hour, Ludwig. I don't know if you want to stop here. You know, I'm happy to take additional questions by email. I'll share my slides.
Sounds great. Sounds great. Yeah, the slides and recording will become available under that link that I put into the chat. And then I just say, let's thank the speaker, Artem, once again. Thanks a lot for coming over, Artem, for a fantastic talk. And see you all maybe next week when we will have Konstantin speaking about some more pre-processing of single-cell RNA-seq data.